Every single day, a mature Fomitopsis pinicola fruiting body expels billions of microscopic spores into the surrounding forest. The traditional aerobiology model assumes these wood-decay fungi rely on passive wind dispersal. The spores are ejected, caught in air currents, and left to randomly settle on suitable dead wood. But modern managed boreal forests are heavily fragmented. Dead wood is no longer an abundant, continuous resource. It is scarce and patchily distributed across vast areas of active timber harvesting. This diagram shows the spatial inefficiency of the wind model. When spores spread radially from a source point, the overwhelming majority fall within just a few meters of the parent fungus. In a fragmented grid, nearly the entire reproductive payload lands in dead zones. To solve for spatial isolation, ecological systems frequently utilize directed dispersal, or zookery. This is the process where animal vectors carry biological propagules directly to favorable microhabitats. We already know certain bark beetles cultivate specific mutualistic relationships with fungi to farm them for food. However, the role of non-mutualistic generalist insects in transporting wood-decay fungi has historically been underestimated. Empirical data shows that saprosilic beetles, insects dependent on dead wood, provide precise, directed dispersal for keystone generalist fungi like F. pinicola. These beetles actively transport living fungal spores across the forest, carrying them on their hardened exoskeletons and internally through their digestive tracts. Proving this required resolving a specific technical conflict. Detecting a fungal DNA sequence on an insect does not confirm the fungus is actually alive or capable of growth. Confirming that these spores arrive alive and ready to colonize wood provides a new perspective on how fungal populations sustain themselves in landscapes altered by human management. Modern DNA metabarcoding is highly sensitive. It can detect a fungal genetic sequence on a beetle's leg, but DNA alone cannot prove that the spore is viable and capable of colonizing wood. Directly testing viability by plating field-collected beetle parts onto standard nutrient agar also fails. The plates are almost instantly overrun by fast-growing environmental molds and bacteria before the target fungus can establish itself. Researchers used a genetic baiting technique, a haploid F. pinicola network with a single nucleus per segment. To mate, two compatible mycelia must fuse. If a living spore from the beetle drops onto the bait, its hyphae will grow and fuse with the network. Once fused, a distinct morphological change occurs. The cellular architecture alters to allow the second nucleus to migrate between adjacent cells. This migration pathway forms a semicircular structure at the cell walls, known as a clamp connection. Researchers isolated heads, wing covers, and feces from wild beetles, placed them directly onto these bait cultures, and sealed them in the dark for 20 days. After subculturing the results, they scanned the fungal networks under microscopes, looking specifically for the formation of those clamp connections. Unlike a passive DNA sequence, the formation of a clamp connection provides observable biological evidence that the spore was viable and active. By requiring the fungus to physically mate with the bait, this method filtered out the surrounding noise of dead genetic material and mold contamination. This technique provided the in-tempo data needed to quantify exactly how effectively beetles transport viable F. pinicola spores across the forest. The first phase of the experiment, quantify acquisition at the source. Under the cover of night, researchers collected 167 beetles spanning 8 different species directly from sporulating F. pinicola fruit bodies. Fungal sporocarps emit chemical volatiles that draw beetles to feed directly on the nutrient-dense spore layers. This chart visualizes the results of the baiting tests on the beetles' exoskeletons. The researchers detected viable spores on the heads of 98% of the insects and on the elytra, the wing covers, at a rate of 97%. Almost every insect walking across the spore layer liberates and stores living propagules on its external armor. But when researchers tested the beetle's feces, they detected viable spores in 83% of the samples. Standard ecological assumptions suggest that the digestive enzymes inside an insect's gut will degrade or destroy fungal spores. To verify these spores were genuinely surviving the digestive tract and not just clinging to the outside of the sample, the researchers implemented a strict verification protocol. They submerged a new set of beetles in saline and ethanol to chemically sterilize the outer shell, then isolated and punctured the midgut and hindgut to extract the internal contents directly. The bait cultures confirmed the discovery. Viable F. Pinicola spores were retrieved from the internal guts of all 3 tested species. Internal dispersal, or endozoochory, protects the spore from ultraviolet radiation and drying out, extending the range the fungus can endure before reaching a new log. Acquiring the spores is only the start. For directed dispersal to succeed, the insect must deliver the living payload to a specific destination. In the second collection phase, researchers placed freshly felled, uncolonized spruce logs in the forest to act as bait targets for saproxylic insects. A newly felled log is the optimal habitat for early colonizing wood-decay fungi, but the competition to establish a mycelial network is high. Over the following weeks, researchers captured 22 different beetle species arriving at the fresh wood. By running these incoming insects through the monokaryotic baiting process, the data revealed 9 specific species actively transporting viable F. Pinicola spores to the new logs. These species are biologically programmed to detect the volatile penicle emissions of freshly dead wood, steering themselves toward the exact habitat the fungus requires. Yet a fresh log presents a physical barrier. The tree's thick outer bark prevents passively wind-blown spores from accessing the nutritious cambium layer beneath. The data highlighted 3 carrying species in particular, including Tripodendron lineatum and Hylobius excavatus. These are bark-boring beetles. To reproduce, they physically excavate galleries through the outer bark, laying their eggs deep inside the vascular cambium. This mechanical excavation solves the deposition problem. The beetle acts as an active inoculation needle, bypassing the tree's defenses and embedding the living spores directly into the wood. Mycologist C.T. Ingold defined 3 distinct stages for successful fungal spore dispersal: liberation from the source, transport through the environment, and deposition at the target. The empirical data demonstrates that non-mutualistic generalist beetles actively facilitate every stage of this model for F. pinicola. Returning to our spatial grid, we replace the inefficient spread of wind with targeted flight paths connecting established fruiting bodies directly to isolated fresh logs. While wind generates the massive volume of propagules required for general coverage, zoochory provides the precision targeting. It ensures that a portion of the reproductive effort reaches viable dead wood. As intensive human forestry practices continue to fragment landscapes, the spatial distances between pieces of suitable dead wood continue to grow. As that distance increases, relying entirely on random wind currents to reach isolated targets becomes mathematically less likely for fungal populations. The survival of the fungus is directly tethered to the survival of the insect vector. If the populations of these specific saproxylic beetles collapse, the fungal transport network collapses with them. Conserving keystone fungi in managed forests requires an integrated approach. Protecting the dead wood is insufficient on its own. We must also preserve the multi-species invertebrate networks that facilitate fungal colonization.